This is Gregor Mendel. Gregor Mendel was an Austrian monk who lived in the mid-19th century, so around 1850 to you and me. Not only was Gregor Mendel a monk, he was also a priest in his monastery. And he was a school teacher as well. But Gregor Mendel's main job at the monastery, and one that he took a lot of pride in, was tending to the vegetable garden. His favorite plant to grow, and one he spent lots of time cultivating, was the simple garden pea. Through his work over many years studying his different pea plants, Gregor Mendel was able to figure out the principles of inheritance, or how traits are passed from one generation to another. For his work, Gregor Mendel is considered the father of modern genetics. Gregor Mendel was lucky in that he chose the garden pea because garden pea is a special variety of plant. Garden peas are special because garden peas can self-pollinate. What that means is that the pollen, which is on the anther right here of a plant, can actually fall right onto the ovary of the same plant. Of course, garden peas can also cross-pollinate, which means that the pollen from one plant can land on the ovary of another plant. Because of this, garden peas are very easy to work with. Because garden peas self-pollinate, the offspring of the garden pea plant have the same DNA and therefore the same genes and same traits as the parent plant. Mendel called pea plants that always self-pollinated true breeding. True breeding plants always produce the same form of a trait. For example, tall plants will always have tall offspring. But pea plants don't always self-pollinate. Like I said before, pea plants can also pollinate other pea plants. Mendel called this crossbreeding. Crossbreeding really happens when plants with different forms of a trait breed with one another. The resulting offspring are often called hybrids because they have a hybrid form of a trait. When looking at pea plants, Mendel actually examined 7 traits, and each of these 7 traits exhibited one of 2 forms. For example, he looked at whether seeds were gray and round or white and wrinkled. Or he looked at whether there were green peas or yellow peas. Or, for example, with flower color, he looked at whether the plant produced white flowers or purple flowers. There were also other traits, such as whether the pods were full or constricted, yellow or green, flowers were on the ends or on the sides of the plant, and whether the plant itself was a tall plant, 6 to 7 feet tall, or a short plant, only about 1 foot tall. What Mendel did to examine these traits was something called selective crossbreeding. In selective crossbreeding, Mendel intentionally bred 2 plants together that showed 2 different varieties of the same trait. For example, 1 tall plant or 1 short plant. We as humans actually selectively crossbreed all the time. A good example is with dogs. When you have breeds of dogs, we can breed 2 breeds of dogs together to make a dog with desirable qualities of both. Or we can keep 2 breeds the same, such as 2 Goldens together, so that you only have Golden Retriever puppies. What Mendel thought was going to happen was that when he selectively crossbred, he would get a plant that showed some characteristics of each plant. For example, when he crossed a tall plant with a short plant, he would get a medium-sized plant. This actually didn't happen, however. When he crossed a plant with 2 different traits, the offspring would always only show one of the 2 varieties of trait. For example, when a purple and white flower were crossed, all the offspring were purple. Or, when tall and short plants were crossed, all of the offspring were only tall. There's also some vocabulary here for you to notice. If you notice, the first generation right here is called the P generation. P stands for parent. So it's the parent generation. In the parent generation of Mendel's cross, he crossed plants with 2 different traits. The second vocabulary term, which is circled in yellow, is F1. F1 stands for the first filial generation. What that means is the first generation of offspring, so the initial children of the plant. Later on, we'll see the F2, or second filial generation, which would be the grandbabies of the parent generation of plants. Finally, Mendel had a word for the trait that always appeared in the F1 generation. He called that trait dominant. In other words, he always saw it, and that trait was dominating out the other trait which disappeared. Mendel then had a second hypothesis. What he assumed was, if he bred the same plant with itself, because remember, pea plants can self-pollinate, from the F1 generation, what would happen? Mendel actually assumed the F1 generation was true breeding, so that if you cross the same plant, for example, the tall plants from the F1 generation, because tall is dominant to short, they would also always have tall offspring. In other words, that short trait would just go away. What actually happened, however, was a little bit different. If you look at the F2 generation, which was created by self-pollination from the F1 generation, you actually ended up with 3 tall plants and 1 short plant. All of a sudden, that short plant reappeared. Mendel tried his experiment over and over again, crossing a tall and short parent every time the F1 generation had all tall plants. After that F1 generation was allowed to self-pollinate, however, he always seemed to get approximately a ratio of 3/4 tall plants to 1/4 short plants in the F2 generation. No matter what he did, every F2 generation was always 3/4 showing the dominant trait, no matter what trait it was, and 1/4 showing the hidden trait, which we now call the recessive trait. So what really happened with Mendel's pea plants? Remember that every individual has 2 copies of the same chromosome, even though they don't necessarily code for the same trait. From meiosis, you'll remember that these are called homologous chromosomes, chromosomes that code for the same trait, not necessarily in the same manner. And every pea plant has 7 pairs of homologous chromosomes. What happened was that in true breeding plants, remember the parent generation was true breeding and always produced the same type of offspring when they self-pollinated, both parents had the same form of the gene. By the way, we call the form of a gene an allele, and each chromosome in the homologous pair had the same allele. If you look below, the tall plant right here has 2 tall genes on its homologous pair of chromosomes that code for plant height. And the short plant right here, I'll make a sad face for him, has 2 short genes in the same location that code for plant height. During fertilization and reproduction, offspring get 1 chromosome in a homologous pair from each parent. So all F1 plants got 1 of the 2 chromosomes in the pair from each parent. Therefore, each offspring, or F1 plant, got 1 tall chromosome and 1 short chromosome. This from the tall parent, and this one from the short parent. Therefore, on the homologous pair of chromosomes in the F1 generation, all F1 plants ended up with 1 chromosome from the tall plant, so 1 chromosome had a tall allele, which is this guy, and 1 chromosome had a short allele, which is this guy. Because the tall allele is dominant to the short allele, only the tall allele gets expressed when both alleles are present. Therefore, all the plants in the F1 generation, even though they had 1 tall and 1 short allele, would all appear to be tall. When the F1 generation self-pollinated, the pollen could give 1 of its alleles, and the ovary could give 1 of its alleles. And due to meiosis, it's completely random which allele it gives to the offspring. So here are the possible combinations of alleles that could possibly be given to the F2 generation. The F1 pollen could give a tall allele. The F1 ovary could give a tall allele, meaning that the F2 plant would get 2 tall alleles and the plant would appear to be tall. The F1 pollen could give a tall allele, but this time the ovary could give its short allele, and the F2 combination would be 1 tall and 1 short, and the plant would still appear to be tall. The reverse could also happen. The pollen could give its chromosome with a short allele, the ovary could give its chromosome with a tall allele, you still end up with one tall and one short, and the plant still appears to be tall. Finally, the F1 pollen could give its short allele chromosome. Same thing with the ovary. The plant could end up with two short alleles, and this time, because there's no tall allele, the plant would actually be short. What you'll notice is the probability of the plant being tall is about 3 out of 4 times, and the probability of the plant being short is 1 out of 4 times, and this actually explains the ratio Mendel saw of 3/4 tall to 1/4 short. To help you really understand this, here are some vocabulary terms that you have to know. The first one is trait. A trait is a characteristic expressed by an organism. For example, your height, your skin color, your eye color, your hair color, whether or not you're bald. These are all traits. They're just characteristics expressed by an organism. The next one is allele. An allele is the variety of gene you have that codes for a version of a trait. Each chromosome has an allele for each gene that it codes for. In this case, the 2 homologous chromosomes have a gene for flower color. One has an allele for purple flowers, the other has an allele for white flowers. They're still homologous because they code for the same genes, but each homologous chromosome actually in this pair has a different allele. To simplify things when we write alleles, we actually write alleles as a single letter of the alphabet. A capital letter indicates a dominant allele. A lowercase letter indicates a recessive allele. A good example is for the tall short alleles in pea plant. We will use T for the dominant allele and little t, lowercase t, for the short allele. It's really important that we use the same letter to indicate the trait. We usually use the letter of the dominant allele. So for example, tall is the letter T. Since tall is dominant, capital T is tall, and lowercase t is short. But we always need to use the same letter for both alleles of a trait. Just one is capitalized, and the other is lowercase. The next vocabulary word is phenotype. A phenotype is the actual trait that is expressed. It's what we can physically see. In other words, the plant is tall, or the plant is short. The next word is genotype. A genotype is the combination of alleles that an organism has for a trait. Remember that because organisms have 2 chromosomes that code for a trait in an homologous pair, they always have 2 alleles that make up their genotype. In the case of the short plant, it has 2 short alleles, so it is lowercase t, right here. A tall plant could either have 2 tall alleles, which is this guy on the right, capital T, or 1 tall and 1 short allele, capital T, lowercase t. The next word is our new definition of dominant. Dominant refers to an allele, and a dominant allele is the allele that is expressed when both alleles are present. In other words, because this plant has 1 tall allele and 1 short allele, the plant is tall. Therefore, tall is dominant. Tall is the dominant allele. The next word is recessive. A recessive allele is the allele that is not expressed when both alleles are present. As you see here, once again, this plant has 1 tall and 1 short allele, but the plant ends up being tall. Therefore, the short allele is not expressed. The next word is homozygous. Homo means same. So a homozygous individual is an individual that has 2 of the same allele for a trait. In other words, 2 tall alleles, like this guy, or 2 short alleles, like this guy. We would call the short plant homozygous recessive, because it's got 2 of the same recessive alleles. And we would call the tall plant homozygous dominant, because it's got 2 of the same dominant alleles. Finally, the last word is heterozygous. Heterozygous is when an individual has 2 different, hetero means different, 2 different alleles for a trait. For example, this plant is heterozygous because it has 1 tall and 1 short allele. In order to predict traits, we use something called a Punnett square. A Punnett square is a box made out of 4 squares. Well, at least the ones we're working with now have 4. We'll get to more complicated ones later. What happens is the alleles of one parent are written across the top, and the alleles of the other parent are written across the side. so that one allele sits across each row and one allele sits above each column. What we do is we cross and meet the alleles in each box so that the possible combinations show all of the possible results. For example, in this one, this would be big T, big T. This would be big T, little t. This would also be big T, little t. And this would be little t, little t. So here's a practice problem. In pea plants, the allele for tall plants is dominant to the allele for short plants. So we know that big T is going to be dominant to little t. If a homozygous dominant parent is crossed with a heterozygous pea plant, what would be the genotypic and phenotypic ratios of the offspring? So let's look at the first parent. Let's do it up top. The first parent is homozygous dominant, which means both alleles are the same and they're both dominant. So he's going to be big T and his other allele is also going to be big T. The second parent is heterozygous, which means he has one of each allele. We'll put him on the side, so he'll have one big T and one little t. We need to know the genotypic and phenotypic ratios of the offspring, so we'll put this in the box. We'll go big T, big T, big T, big T, see where they meet. This is big T, that's little t, so we'll go big T, little t, and then big T, little t. And what we have here for our genotype ratio, remembering that genotype is the combination of alleles you have 2 out of 4, big T, because the ratio we use a colon, to 2 out of 4, big T, little t. It's also okay if you express this in percentages. So for example, I could also write that this is 50% big T, little t, and 50% big T, or I could also do 1 out of 2 if I wanted to simplify the fraction. For a phenotypic ratio, this is what we see. And since all of the plants have a dominant allele, we could say this is either 4 out of 4, 100%, or just simply all tall. Here's another practice problem. This time we're going to use a different trait. We're going to use seed color. And in pea plants, the allele for yellow seeds is dominant to the allele for green seeds. So we're going to use big Y for yellow and lowercase y, which I'll draw a little differently, for green. This time we have a heterozygous yellow plant. Because it's heterozygous, it has one of each allele. We'll put him up top, so he's got big Y and little y. And the other plant is green. Now, we don't have the genotype here, but we do know that green is the recessive trait. In order to be recessive, you must be homozygous recessive, so therefore he has to be little y, little y. Let's do the genotypic and phenotypic ratios. Once again, we use our Punnett square. Big Y, little y, little y, little y, big Y, little y, and little y, little y. And once again, we get 50% big Y, little y, to 50% little y, little y. And what about phenotypes? This is a little different. This time, these plants on the left are yellow, so we get 50% yellow to 50% green. And that's it for genetics. If you can get this, you're off to a good start.